from you and your place. We have two speakers tonight. Dympna Cullen will be speaking first and speak for 20 to 25 minutes, and then we'll have a Q&A session immediately after that. And then we'll move on to our second presenter tonight, Ben Stepkovitch, who will speak for 20, 25 minutes and have a Q&A. Now, these are rough times, but that's generally the way it will go. So during the presentations, I ask you all to keep your microphones muted. For the Q&A sessions, you can ask questions by virtually raising your hand in the reactions box or using the chat function to send a message to the off host that you want to ask a question, or you can type your question into the message and it can be read out for you. Please mute your microphone after asking your question. We'll then move into off general business items. The meeting is expected to take 90 to 100 minutes. You're free to leave the meeting at any time up until the meeting is closed by the host. You can do this by clicking Leave Meeting. So now to our speakers, we'll get straight on. Our first speaker tonight is Dympna Cullen. She will tell us about the research to determine the attributes and connectivity of ecological refuges for the crest-tailed Mulgara following a prolonged period of drought. Dympna is a PhD candidate at UNSW and off research grant recipient last year. She works as a Casual Senior Field Ecologist for the Australian Wildlife Conservancy on various translocations and surveys, and as an independent wildlife biologist on various projects. Prior to commencing her PhD, Dympna worked and did research at, I think it's the AWC's Scotia Sanctuary, and researched koalas on Kangaroo Island. So with that brief introduction, I'll hand it over to you, Dympna. Thanks very much.
Thank you. So I'd like to just start by acknowledging the traditional custodians of the lands across Australia on which I work and do my research. And I'd like to acknowledge that sovereignty was never ceded and pay my respects to elders past and present, and extend that respect to any First Nations people here today. So I'll start my talk by just giving you a bit of background about myself and brief overview of a couple of the other projects that I've worked on, and then I'll talk a bit about my PhD research. So I did a Bachelor of Science and Bachelor of Arts at Monash University, and then I moved on to a Masters of Wildlife Health and Population Management at the University of Sydney. And I did my research in collaboration with the then Office of Environment and Heritage. And we were looking at a population of koalas in the Southern Highlands of New South Wales. And while koalas are commonly considered to be dietary specialists, they've successfully inhabited a geographic range that encompasses a large variation in eucalypt species, as well as climate. And so the Southern Highlands population hadn't been studied before. So we wanted to know what species koalas used in the area and to identify the composition of eucalyptus species required for habitat conservation and restoration. And so the aims of my research were to establish what tree species koalas were selectively using. So the tree species that were disproportionately used to their abundance in the koalas home range. And also, so we collared 20 koalas with GPS and VHF transmitters and radio tracked them over 8 months, recording the species and size of the trees that they were using. I then completed tree species surveys across their home ranges to compare the abundance of tree species in the home range with those that the koalas used. And we found that koalas use trees with a larger average trunk diameter and underutilised small trees. And this has management implications and conservation implications because while a patch of potential habitat may contain a high proportion of regionally identified preferred habitat trees, The value of younger or single age class forests may not hold the same value as that of old growth or mixed age class forest. And so while regeneration of habitat is an important component of koala conservation moving forward, we really strongly argue that it's not a suitable mitigation measure for destruction of habitat. And we found that koalas used 20 tree species all up in the study area. And when we divided that into 3 regions based on soil type, we found that koalas were selectively using 3 tree species in each of those areas, and that they were unique to each area. And the number of tree species used and that the koalas selected different tree species in these areas shows koalas' adaptability to variation in habitat, even within a small geographic range. And at the time of the study, only 1 tree species that we had identified was recognised in koala habitat protection legislation. And so subsequently, the results of this study have been used in a review of habitat protection legislation, and we were able to get 6 more tree species added. And then following on from my master's, as Kim mentioned, I worked on Kangaroo Island for a season, managing a team of koala catchers on the sterilisation program. Koalas were introduced to Kangaroo Island in the 1920s and quickly became overpopulated, with overbrowsing reported in the 1960s. And koalas don't regulate their breeding in accordance with food availability. So there was concern for starvation of the koala population, with continued long-term impacts on eucalypts from overbrowsing. And in 1997, the koala management program was implemented. And the program has involved habitation programs. And while the idea of controlling koala populations has and is contentious, as a nation, we're pretty protective over our iconic koala. The outcome of not implementing effective management in these translocated populations can have some pretty devastating outcomes. Koalas preferentially eat certain tree species to the point of complete defoliation of trees and mass starvation, which we've seen in areas like Cape Otway. So our job was to catch koalas for surgical sterilisation and re-release. And for those that don't know how to catch a koala, this involves first walking around and trying to spot a little grey blob up in the tree. And I can tell you this is much easier to do on Kangaroo Island with the density they have than it is in the Southern Highlands with their low density population. And then we harness up and climb up the tree to near the koala. And we place a guide rope on using a long pole, which helps to direct the koala safely down the tree. And then you wave a flag over their head, and they don't really like that, so they start moving down the tree to a waiting ground crew who can put them in a bag, or sometimes we can actually bag them in the tree as well. And from there, I moved to an area with very few trees to climb and definitely no koalas. Although it did take me a long time to stop automatically scanning every tree for koalas. And I started working for the Australian Wildlife Conservancy. I spent 6 months at Scotia Sanctuary in the Mallee of southwestern New South Wales. And then I moved up to Newhaven Sanctuary in the Tanami Desert in the Northern Territory. And I was up there for a few years. And Newhaven is just absolutely spectacular. It has a really fantastic diversity of particularly reptiles, and it's also known for its bird life. And during my time at Newhaven, I was lucky enough to be a part of the construction of the 10,000 hectare feral predator and rabbit exclosure, and also to be able to reintroduce the rufous hair wallaby, or the marla, back into their historic territory. Marla have suffered a pretty similar fate to most arid zone critical weight range mammals, being severely impacted by the introduction of cats and foxes, and they came very close to extinction. The last wild population of marla, which I believe was 22 individuals, was taken from the Tanami Desert in 1991 and placed in a captive breeding program. And today marla only exist on one island and in a handful of fenced reserves. So to have the opportunity to be a part of the team that returned them to their historic range and to an area that where they're very culturally important for the Warlpiri traditional owners, it was a real highlight of my time at AWC for me. And while at New Haven, I also got to work with brush-tailed Mulgaras up in the right-hand corner there. And this would lead me on to doing my PhD with their close relative, the crest-tailed Mulgara. So crest-tailed Mulgaras, also known as Amperdas, they're a small arid zone carnivorous marsupial, and they weigh about 60 to 180 grams. And because quolls have gone extinct across most of the arid zone, Amperdas are currently the second largest native predator under dingoes in these ecosystems. So they play a pretty important role as a mesopredator, and I can tell you they have the attitude to match. They have some pretty amazing physiological adaptations to reduce their energy expenditure, including going into torpor in their burrows and dropping their body temperature to around 10 degrees. And then they crawl out to the entrance of their burrow during the day and use the sun to warm up. So they don't have to use that metabolic energy to warm up. And so by using these kinds of strategies, they can reduce how much energy they need to use to find food, which can be pretty important in the arid environment where things are pretty scarce. And you can see their little beautiful feathered crest on their tail, which is one of their identifying features. The map there shows their historic range in grey and their current known range up until 2018 in red. And you can see that they've suffered pretty significant range retraction since European invasion. They're listed as vulnerable nationally, endangered in South Australia, and presumed extinct in New South Wales. And because there was a lot of taxonomic confusion over the years within the Dasycercus genus, we really know very little about their biology or ecology. So Amperdas are a Dasyurid, so they're a carnivorous marsupial related to Tassie devils and quolls. Australian Dasyurids, they have quite a broad range of life history strategies. Some have short lives and produce lots of young, and some opt for longer lifespans with more opportunities to reproduce. And these life history strategies are driven by adaptations to both predictability and also abundance of resources in different environments. And Australia's arid and semi-arid zones are characterised by unpredictability of resources. They have huge seasonal and annual climatic variability and resulting prolonged periods of low resource availability. And arid zone mammal species fluctuate hugely in abundance, either booming during brief periods of high resources or fluctuating in abundance seasonally. And the primary driver of population change is considered to be rainfall. And you can see the bottom photo there, just to give you an idea, this was one of my study sites in 2019 when I started my PhD. And particularly for those that are familiar with the desert, you can see there is just absolutely no vegetation there. It's just completely bare. So I started my PhD wanting to understand how Amperdas survive in these really tough times with prolonged low resources. Some of my questions are looking at whether they shift their diet from periods of wet to dry, whether introduced predator scent affects their foraging behaviour. I'm also looking at population genetics to help us understand their current and historic distribution and see if that can give us any clues as to whether they retract into kind of core refuge habitats. And the two that I'll sort of talk about today are looking at what's driving their ongoing range expansion and whether resource availability alters their reproductive strategies. So we knew from Rhys Pedler and his co-authors' work that following releases of rabbit hemorrhagic disease virus, which is also known as calicivirus. Starting in 1996, rabbit numbers plummeted, and you can see that from the blue line. And rabbits were supporting inflated numbers of introduced predators, and so subsequently the cats in the yellow and foxes in green also decreased. And at the same time, between 1995 and 2014, amperdas were actually expanding their range from the small yellow sliver, which you can see there, where they'd been restricted to since the 1970s, into those blue areas. And since then, we've observed them continue expanding their range to the southeast and into that green area. And this is during a period of prolonged low rainfall. So to give you an idea of how bad things have been, the green square there highlights our area of interest. And so the dark blue shows very high rainfall above average, and the dark red showing well below average rainfall. So sort of in that 10 years of the millennium drought, we had quite low rainfall until we hit the break in the millennium drought in 2010, 2011, where we had a couple of years of decent rain. And then since then, things have been pretty bad again, with just 1 year of a bit of good rain. And then you can see in 2019, when I started my PhD, things were pretty rough. And we've had a little bit of rain since, but the country had a lot to recover from. So I wanted to know a bit more about amperda population dynamics and reproductive strategies, both from a management perspective, given that they're a threatened species, and also to try to understand their range expansion. So if we accept the rainfall is indicative of resource availability, we looked at whether low resource availability would result in decreasing abundance, changes in the length or timing of the breeding period, whether the the proportion of females captured with pouch young would decrease, and whether average litter size would increase. My survey sites are in the Strzelecki Desert at the southeastern extent of their current known range, which you can see again there in red, with their historic range shown in the grey again. And the black dots are our trapping sites. And down the bottom there, you can see my portable home office where I do my data entry in the afternoons between checking traps. So we set about 9,500 Elliott traps across that area and recorded their reproductive status based on their pouch condition and their testes width, their age based on their weight and their reproductive status, and their body condition based on their tail width and their weight. And with ours and previous data, we had capture data from 2015 to 2021. And these sites had started to be trapped in 2011, but they only started to record amperdas in 2015 as they expanded into that area. So looking at our first question of abundance, we found that they were first recorded in November 2015, and captures of adults in pink and juveniles in yellow show that the population is generally increasing. Across years, we detected pouch young from May to November, red in the left-hand figure, and an increase in juveniles from August to March in yellow in the right-hand figure. And both of these indicate a single annual winter breeding period. And no females that were caught on multiple occasions showed signs of attempting to have a second litter in a single year. And the data was a little bit sparse to identify if either the timing or the length of the breeding period changed from year to year, but the length of the breeding period does suggest that there may be some limited flexibility in the timing of breeding, either as a response to environmental variables or with females entering a second estrus if their first mating was unsuccessful, which we know to be the case in brush-tailed mulgaras, so it's highly likely in crest-tail as well. And while a static breeding period may seem counterintuitive in a resource-poor environment and result in inflexibility to respond to environmental conditions, for most Dasyurids, unpredictability of resources usually drives a bet hedging strategy of longer lifespan, larger body size, a longer breeding period, and repeated breeding over several years. And all of these increase the potential for reproduction to coincide with a period of high resource availability. So for Ampertas, this results in longer timeframes over which the populations fluctuate compared to smaller placental species like rodents, which exhibit shorter boom and bust cycles. And females showed evidence of breeding in all years from 2017. The yellow shows the proportion of total adult females captured over those winter to spring breeding months between May and November, with 67 to 100% of females showing signs of breeding. And Ampertas can have up to 8 pouch young in a litter. You can see they've got eight teats there. And in years where litter size was recorded, average litter size was 6.5 plus or minus 1.8 standard deviation, with a minimum of 1 and a median and maximum of 8. And it's interesting that, as you can see from the left-hand figure, average litter size showed a general increasing trend over that 5 years, and the majority of litters were a full 8. And this was despite all adults we captured being in poor body condition. And this is particularly interesting because in general, arid zone Dasyurid species are known to rarely produce full litters, which has been attributed to low food availability. And now there's a lot going on in this graph, but don't worry, it's kind of just bringing everything that I've just been talking about together. So as we saw in the earlier slide, the pink and the yellow bars indicating the captures of juveniles and adults show that the population's been increasing in abundance, and as we saw, increasing in occupancy as well. And this is despite low cover. So as you can see, the orange line shows bare ground increasing, which indicates that cover has been decreasing. And then add to that you've got prolonged low rainfall, which you can see in the blue line there. So we can't say that the rainfall and the cover is definitely driving population dynamics, but what's interesting is that given these conditions, this is not what you'd expect to be happening. And the lack of cover and rain doesn't seem to be hampering Ampertas establishing in new areas and building up in numbers. So to try to understand what's driving reproduction and population dynamics, I'm currently modelling our results from this site and other sites further west in relation to environmental variables, predator and prey availability, and also whether their diets shift their genetics and predator naivety, like I mentioned earlier on with those other questions of my PhD. So what does all this mean? Ampertas are physiologically adapted to these extreme conditions. They have very low water requirements, and they reduce their metabolic energy expenditure through the use of torpor and sunbaking. They're also dietary generalists, and from some of the early results of my dietary work, it looks like invertebrates play a really important role in sustaining Amperta populations. We know from Central Australian deserts that during periods of drought, when rodent and reptile numbers decline, invertebrate populations have been able to withstand the increased predator predation. So being so wonderfully adapted to extreme conditions and able to reliably produce large litters, it's likely that Ampertas can really take advantage of the reduced predator pressure that's resulted from a combination of reduced rabbits and the drought conditions. And so, like I said, I started my PhD looking at how Ampertas survive in these really harsh conditions, and I've kind of shifted to looking at why they're thriving. So I'd like to say a huge thank you to the Oatley Flora and Fauna Conservation Society for supporting my research, also to my supervisors, my co-authors and collaborators, other funding bodies, and also to all of the volunteers who've come out and helped me with their time to get all my fieldwork done. And so, is there any questions?
Thanks, Dympna. That's fantastic. It's really interesting. Such cute little animals, and they're very hardy, it seems.
Yeah.
So, any questions out there that people want to lob in? There's nothing in the chat
ask one,
that
Kim,
I can see. Deb,
Kim, I
yes?
could ask one if that's all right.
Okay.
Dympna, I'm fascinated. It's a great talk and it's amazing thinking they can survive. So how would you describe the habitat of the crest-tailed Mulgara if there's no vegetation required?
Yeah, so they live sort of they live mostly on the dune tops. So there's sort of there's usually a sort of sparse number of shrubs, so things like acacia ligulatas, the sandhill wattle, I think is the common name, and hot bush and things like that. So there is usually sort of some sparse vegetation there. And then they do go down into the swales, so the areas between the dunes where there's sort of lower vegetation to hunt. But they tend to burrow sort of up in that dune area. But from what we've seen with crest-tailed mulgaras and also with brush-tailed mulgaras, they sort of seem to use the areas in between the vegetation a lot more than they do actually under the. You know, we see them and think they're this small little animal that's going to act like a prey species, when they sort of seem to act more like a predator. You know, they use that in between to hunt.
Yeah, okay, terrific.
Dympna, Garth Nicholson here. I want to ask you, Garth Nicholson, can you hear me?
Yes, we can,
Yes,
Garth.
yep.
Go ahead.
My father spent his life working on population dynamics in blowflies, and he had looked at how they respond with the chaos equation to, in an environment that didn't change, their populations fluctuated wildly. And he's tried to work out what was behind it, and his idea was it was their biology that did it. And I was wondering whether your interpretation of what you found fits that, but it's certainly more complicated because you're looking at variable changes in the environment which are simultaneous with the numbers that you're measuring.
Yes, exactly. And that's sort of the hard bit to tease out is, like I sort of said, you know, we're observing these trends, you know, which are correlated with these environmental variables that we're observing, but we sort of I need to do some more work on the modelling to really kind of tease out what's driving it and what, you know, whether that's just sort of something that seems to be happening at the same time.
It seems to be that when their numbers are down, they work to get their numbers back up again. And you've more or less shown it with the increase in litter size. And it'd be interesting to know what was behind that.
Yeah, that's it. Yeah, whether they actually do increase that, the number that they have.
How did they know they had to do that?
Yeah.
It's a bit like supposedly in war, human populations increase.
Yeah. Yeah, well, they're at the invasion front, these Mulgaras, so I think they know they've got to keep repopulating.
That's about it, yeah. Okay, other questions out there?
Vicky's asking a question. What is the lifespan of the Mulgara?
We don't know exactly with wild Mulgaras. It'd be around three years or so. there was a couple of captive Mulgaras which have lived up to 5 years in captivity, but you can kind of assume they might not all get to that age in the wild. Sorry, just turn that radio down.
Yeah. Can I ask a question? What's their prey? What do they normally eat? Isn't it?
Yeah, so they'll basically try and eat anything up to the size of like a house sparrow, but the majority of their diet is made up of invertebrates, and they do eat basically anything they can get their hands on, but they do eat reptiles, they'll eat mammals, but yeah, the majority of their diet comes from invertebrates. Yeah, I was actually, oh, sorry.
Oh.
Yep.
I have another question, if it's all right. Have you found anything about their movements and home range? Do they have a set home range, or do they have what, you know, some desert dairy Dasyurus have kind of movable home ranges where they don't particularly stay in one particular spot?
Yeah, so they do have established home ranges. I put VHF collars on some of them and radio tracked them, and they had quite established home ranges. And I was radio tracking them over the breeding season as well. So I wasn't sure if maybe, you know, the males particularly might move a bit more, but they still had reasonably small home ranges than what I would expect. But then I think that, so the individuals that I've been catching sort of right on that invasion front, they tend to be subadults. So I think the subadults are the ones that then, you know, disperse out of those home ranges, which is, yeah, what happens obviously with a lot of species.
Dympna, can I ask another question? This is all within the fenced area, isn't it? They've removed the main fox predators. Is there any other predators there, the dingoes there?
So most of my work is, yeah, in the non-fenced area. So these are wild populations. So I can show you, see if it'll go back to that map. Oh, sorry, a million slides. There we go. So it's kind of hard to read because I've blown it up from the paper, but that plain, so the black line there, that's the dingo fence, the dingo barrier fence that separates New South Wales and South Australia, and northern South Australia from southern South Australia. So in that shaded area, you have dingoes present, and then in the non-shaded area, there isn't dingoes. There's basically no dingoes. So yeah, it's quite, yeah, it's interesting. So over that side, there's sort of, on the whole, maybe a few less cats and foxes than the New South Wales side. But there's still definitely some cats and foxes over there, and dingoes as well. So
you don't have data on the fenced area versus the non-fenced area?
So in terms of the Wild Deserts project, so just in the corner there of northwestern New South Wales, there's the Wild Deserts project where I am now. There's 2 fenced reserves in there, and they have removed all of the cats and foxes and rabbits from in there, and have actually reintroduced Mulgaras in there as well. And so, yeah, they're doing some work on that, looking at inside the fence versus outside the fence. And they're so far, the Mulgaras in the fence are pretty fat and happy.
One more question. Anybody?
Can I ask a question? It's Karen.
Sure.
We'll give you a video as well there. I was wondering about 2 things. You're carrying on from the predators. How about native predators like snakes, goannas, or the birds, the raptors? Is there any data as to what are their natural animals that might predate on them?
Yeah, so yeah, I guess kind of thinking about their sun-based behaviour, you know, is that they're coming out of the city during that time, and that they are potential prey for diurnal birds of prey, you know, brown kelpies and things like that. So it is quite a risky behaviour. There were interesting questions there to look at. And then as far as native predators, Dingo is to date on them. I've got a video on camera digging up a Mulgara burrow and then looked at wood. And I assume that would eat them. We don't know, but I have done an experiment recently looking at predator behaviour in response to both and native predator scents, so dingo and wommer being some of the native predators, and then I had Quoll in there as well, which in that environment for a long time. Haven't analysed all that data yet, so unfortunately I can't give you any conclusions from that, but I'll
Okay, the other part of the question was, you may have talked about this, we were a few minutes late, but are they a social creature? Do they have burrows where they're communal, or are they, you know, just in very small groups, or as solitary and only meeting up at breeding?
Yeah, so they're primarily solitary. Sort of on occasion get that there's times when either a male and a female, or at times sometimes a male would actually go down another male anymore. He'd move somewhere else. So on the whole, they mostly just meet up, I think, during breeding season. But I have found a few females that would share burrows for a little bit longer. And it seems that maybe the females, they don't move as far from sort of their natal burrow. And then, so, you know, those females may have been related females. I can't say for certain, but that sort of seems to be how things work.
Look, thanks for that. I think we'll need to move on to our next speaker. Dympna, happy for you to stay around, or you can leave if you want to. It'd be nice if you could stay and participate in the next presentation. Would that be all right?
Sure. Am I still sharing my screen with you?
Yes, you can stop sharing your screen, and
I'm gonna find where's the stop? Hang on, sorry. Oh, there we go. Sorry, it's vanished on me. I can't find the other bit that I need to.
Down the bottom, the green.
It's improved.
Oh, good.
Scared everyone else away.
They're down the other room. That's
I
not.
hijack it. Does that work if I hijack it?
Sorry.
Share it without
it's up.
permission?
Yeah, there we go. So my second screen. Is that better?
Okay.
Yep, we're on to Ben's now. And yours is gone. So, okay, I'll just introduce Ben, our second speaker. Ben Stepkovitch will present his findings from investigating both positive and negative effects on the ecosystem of reintroducing a native predator, the western quoll, inside the Arid Recovery fenced reserve, South Australia. Ben is a research grant recipient studying for his PhD at UNSW Centre for Ecosystem Science, School of Biological, Earth and Environmental Sciences. He also studied urban red foxes in Sydney for his Master of Research. And Ben is over in South Australia at the moment. Dympna was in Central Australia. Ben's not too far south, or relative terms, a long way south, I think, actually. Anyway, over to you, Ben. Thanks very much.
Great, thank you, Kim. Yeah, so thanks everybody for joining us tonight. I'm coming here from Cookatha country here in South Australia, so I'd just like to acknowledge the traditional owners of the land that I get to work on. Yeah, so as Kim mentioned, I'm looking at what happens to an ecosystem when we put a native predator back in or inside a fence, one of those fence reserves in Australia, which I guess is a closed ecosystem where things are mostly trapped inside and they have to live with each other. So I'll just go through a bit of background and then, talk about what I've kind of found so far in my work. So in terms of predator reintroductions across the world, some of the most famous ones are, like the reintroduce the wolves to Yellowstone. And so a lot of our understanding, I guess, comes from of what happens when you reintroduce predators into areas where they've been lost, comes from these big iconic reintroductions where a lot of work has gone into, a lot of research has gone into. But in terms of reintroducing predators to defence reserves, most of it's pretty uncommon compared to the unfenced areas, but most of it's happened in South Africa where ecotourism has driven, the reintroduction of lions and African wild dogs and cheetahs into the fence reserves for ecotourism. So based on the Yellowstone wolf reintroduction, what they thought they observed was before the wolves were reintroduced, there were lots of more herbivores around, and that led to kind of impact to the vegetation below them. But after the wolves came back, they observed, they kind of kept the herbivores under control, the elk and other deer species around, which made changes to the vegetation. And because that ecosystem is so well studied, everybody kind of assumes now that when you put a predator back into an area that things like that always happen. But unfortunately, it's not always the case, and we're lacking a lot of, I guess, research into a lot more people who've done reintroductions and worried about what happens to those specific animals. Do they survive or not, the predators? And instead of continuing on researching what happens after you reintroduce them for a few years, because some of these things do take a bit of time. So my field site is another fence reserve in Australia, in South Australia, the Arid Recovery fence reserve. So it was built in the late 90s, and between 1999 and 2001, 4 reintroduced marsupials were put into this reserve. The greater bilby, the burrowing bettong, which is a small type of macropods, so related to the kangaroos and wallabies, the shark-bay bandicoot, and the greater stick-necked rat. So that's a rodent. And those, the last of those 3, they're all extinct on the mainland. They've only found in offshore islands, except for where they've been introduced to these fence reserves, whereas the bilby still persists in some areas of Australia on the mainland. So for 20 years, those species have lived inside the fence reserve. The Arid Recovery Reserve is about 120 square kilometres. So this bottom half, the 60 square kilometres, the bottom is where all the feral free bit where those species have been living 20 years. And so in that time, they've started to notice with some of them, they get these problems because there's no predators in there, that they start to become more overabundant than they would in the wild, but also because nothing's eating them, nothing's forcing them to be smart. So all the dumb ones kind of grow up and breed, and they lose a lot of their anti-predator responses. And so the top half of the reserve has started, it's being used as an experimental paddock in the last 10 years or so. They've started to put some of these animals into paddocks with a controlled amount of cats. And shown that by exposing them to cats and getting them a couple of generations to survive with cats, you start to see changes in their behaviour and things, which might help them one day live outside a fence one day.
They reintroduce cats.
That's kind of, the future of where all these fence reserves are heading, is to try and train up these animals to live outside fences one day. That's a big part of it. So in 2018, I was an intern at Arid Recovery, and when I got there, the bettong specifically were at the overabundance. There were thousands. They were everywhere. When you have pipes and things, causing a bit of trouble to us, but also overabundant. They were the other. They're a social animal. They live in groups, and they get up to little fights and about other interns capture this fight. But right now, been a few reintroduced to other parts of Western Australia, including as well the Flinders Ranges, where they've successfully reintroduced in there as well. And so, the thought being by conserving the quoll, translocating the quoll into the arid recovery. They're a threatened species themselves. Can we save another threatened carnivore while also preserving our other threatened species that live inside arid recovery? And hopefully they can help to address some of those issues I mentioned with the overpopulation and animals losing their predator awareness inside French reserves. So in 2018, 12 quolls were flown across from WA. They were caught in the wild in WA and flown across that day to here in Roxby Downs. And Mel was the research officer who studied them for a year after their reintroduction. And in that period of 2018 and 2019, Mel found that the ones that stayed inside the reserve, they started to breed and establish a population, but she also did a lot of their diet in that first year. And she found in over half of their scats, they were eating beddongs, which was a good sign because there was a lot of them around. So they already looked like they were starting to tick that box of starting to address that issue. But they also, the Western quoll's a generalist predator. It's an omnivore, it's a scavenger. So they will eat insects and things, anything they come across, including these carcass dumps we put in the reserve as another source of food for them, just in case they decided to go to their taste for all the endangered animals in there. We provided them some free food to help hopefully save some of them a bit. So that was in the first year that Mel was here, and I've kind of picked up in 2020 when I started. So there's a little bit of gap between when Mel left and I left. But the cool thing about arid recovery is, like I said, that bottom half of the reserve here is all feral free, but there's one of those paddocks that has been kept quoll free. So we have 2 treatment areas where we can compare all those bilbies and beddongs and stuff living without quolls in the main exposure to the ones that have been exposed to quoll since 2018 in the rest of that feral free area at arid recovery. So my job is to kind of look at what effects have been happening, what changes to the ecosystem and those animals have been having since the quolls were reintroduced. But to complicate things, in 2018 and 2019, we went through a pretty bad drought. And so not just, I guess what we're looking at now is the effect of just quolls, but also a lot of the species got knocked down because of the drought as well. So the drought kind of broke in 2020, but it's slow. Things are slowly taking time to grow back. So some of the stuff I'm looking at is going to take a little bit of while till we can work out what effects are just due to the drought breaking, what are the effects are still to the quolls. My project is looking at the positive and negative impacts of reintroducing these quolls, like I've touched on. So I'm looking at kind of 3 main areas. The positive impacts being, by exposing these animals to a native predator, can they regain some of those anti-predator responses that they've lost? Can the quolls successfully regulate those overabundant herbivores like the beddongs and bring about changes to the vegetation to bring it back to what it kind of should be when it's in balance? And then assessing any negative impacts, which is the longer the quolls are here, are we going to start to lose some of those species? Are the quolls going to hunt some of those other endangered animals inside the reserve to extinction within inside the reserve? So I've been pretty lucky. I'm from Sydney as well, but navigating the lockdowns and getting out here to South Australia when we're not in lockdown and spending a lot of time out here doing a lot of field work. It's very pretty reserve, very different to where Dympna works, a bit more flat, but some cool, lots of dunes and things. So part of my project's not just looking at quolls, but it's about monitoring some of those prey species that quolls might be eating. So in 2020, when I first got here, one of the first things we did was capture a bunch of the potential prey animals of the quolls in both those 2 treatment areas, so the areas without quolls and with quolls. So we caught a bunch of beddongs and bilbies in both those paddocks, and the beddongs you can put a radio collar on. The bilbies don't really have a neck, so that we need to put them on their tail, so they get a little transmitter trapped to their tail, so they have to do bilby hairdressing and shave their tail to make it nice and smooth, and they get a smaller transmitter, which because it's lighter, it doesn't last as long, and it can kind of fall off on its own. So I'll get to that in a bit. So the first of those things, those research aims that we were talking about, was looking at what happens how does the behavior change with some of those animals that are living with quolls. So I set up a bunch of experiments. There's food trays, we put food inside them, track them to the beddongs to their burrows, and put out these food trays near them and film their foraging behavior. And so we started doing it just with no kind of stimuli there, and could try to pretty much it all revolves around looking at how vigilant they are compared to non-vigilant. So vigilant, looking over their shoulder when they're feeding, compared to just putting your head down and going for it and in full quickly. And so we did that, and we also started to add in some different stimuli to stimulate some of those predators that they aren't exposed to, things like cats that they haven't been exposed to within the quolls, and then rabbits and water as well as kind of like non-predatory things that are controlled. So I did 3 rounds of experiments putting a stimuli next to the trays, which included scented towels from those animals as well as their scats, and then also placing this army of scary models one at a time at those food trays and seeing how the bedongs reacted to the big scary cats and quolls. And in terms of the results, I did a lot of I took a lot of like effort and stuff to like do all these things, especially with the all the stimuli and stuff. But like in terms of the bedongs, they didn't really react to the ones that are exposed to quolls, didn't really react to the quoll scent or the quoll model specifically. But overall, the bedongs living without the quolls, they were overall less vigilant. They spent more of the time with the head in the bucket, just gorging themselves down. Whereas the bedongs that were living with the quolls, we found consistently across all the different types of stimuli, they were looking out for themselves a bit more, stopping to look around when they were eating, which is pretty cool. It's it doesn't necessarily mean that's going to help them right now survive outside a fence one day, but it's helping to document that by exposing the predators again, you are starting to change their behavior to back what it should be. And there's lots of other research going on here about kind of what drives those changes, but that's someone else's job, not my problem. And that's to say as well, even though they didn't respond negatively to the quoll models, not every interaction between a quoll and a bedong is a negative one. Some of the Karen traps have caught them playing nice together, but it goes both ways as well. There's some cheeky bedongs that like to attack the quoll model. So that's a quoll model there getting harassed, and then this one comes back to finish it off and steals them. And I've got another slide here just of bedongs doing silly things. Poppy one's checking out this bucket, popped in for a squiz, and tips it over, and then this bedong down here stole one of my towels. He runs off with it, ruining the experience for that night. So they are quite cheeky. They do get up to a lot of mischief. So yeah, that's the behavior stuff. And then the other thing I'm trying to kind of document, which is I guess to try and parallel what's been shown about like the wolves in Yellowstone, now that they're in the vegetation below them, the bedbugs that grow into another browser as well. So up until 2017, there've been lots of surveys done by other staff at the reserve documenting the level of browsing damage, the kind of the portion of sticks and stuff that have been chewed on and eaten in the reserve. And again, at the height of that bedong overabundance before the quolls came, before the drought happened, in all areas of the reserve, they noticed there was a high level of browsing damage due to the bedongs. And then I restarted that. I had a bit of pause from that while they were doing sorting out the drought and stuff. And then I restarted those surveys in 2020. And since that time of the drought, the bedongs in both those areas with and without quolls have dropped. And there's less bedongs where the quolls are, but there's still much less bedongs without the quolls. So that kind of shows a bit of effect of drought. So right now, there's not a lot of browsing going on, so I can't really show any changes. But as time goes on and hopefully the bedongs in this area without the quolls get back to where they were, hopefully we'll start to see a bit of a change in the vegetation, which would be another cool thing to show all the predator people and get them excited about that's going on here at the reserve. And then, yeah, the last kind of research areas, again, looking at, how the quolls, what threats do they pose to some of those endangered animals. So we radio tracked 15 bedongs in the main exposure for a year and 14 that were living with the quolls for a year. And we tracked them every couple of weeks while we're doing all those experiments and things to compare their survival. And the ones without quolls, we lost at least one, found at least one dead one in that time, and a couple went missing. And it wasn't due to any other predators. It was just simply times are tough, and a lot of them kind of just dying of natural causes because they're in poor conditions, not a lot of food around for them. But the 14 that were living with the quolls, we lost one of them, but it wasn't due to a predator. It was due to poor condition, and there was no signs of attacks or anything on it. But what's interesting is those 14 that we caught in 2020, after we collared them, we didn't catch any new beddongs or beddongs without collars, or see any beddongs without collars for about 6 months on any cameras, which until they started to breed again once there was a bit of rain. And so that, I'm 99% sure that those were the last 14 beddongs alive in the quoll exposed area of the reserve, which is pretty cool because it shows that they've gone through a crash, and hopefully the ones that are surviving, they survived quolls for a year, which kind of shows the quolls moved to something. They were eating them at the start when there was lots of beddongs around, and as they dropped, they started to move on to other things. They didn't hunt them to extinction, which is the kind of what cats and foxes can do. They can kind of cause local extinctions of the animals. So that's pretty cool so far. And in terms of the bilbies, like I said, they're a bit tricky to catch and monitor and stuff because their transmitters are small and they can fall off and they don't last as long. So in the main, none of them died, which is what you expect. But in the quoll exposed areas, none of them died, but we lost a lot of the transmitters. So that, again, they're a bit tricky to track and stuff. And we tracked some of them for a couple of months, but if anybody's ever seen a bilby in the wild, and here at the reserve, instead of setting cage traps for them, we actually go and chase them and drive around with spotlights and chase them to catch them, because it's a better way to catch them. They're pretty quick, and so if I was a quoll, I wouldn't bother chasing a bilby down. I'd go for something a bit slower and stupider. So I don't think the bilbies are at risk of being hunted down by quolls. And the quoll diet, so like I said, Mel kind of collected a year's worth of data when the quolls were first year, and they had the whole pick of everything in the reserve. And then since that time, we've still been collecting scats, but there's been a bit of a backlog, and I'm picking that up now and going through it. So I'm going to, in the lab back in Sydney, go through all this quoll poo and try and paint a picture of what's been happening since the kind of drought's broken and things. So in 2020, when I first arrived, like I said, a lot of those animals, the beddongs and stuff, had crashed, so they were eating other things, which I'm assuming were increased reliance on the carcass dumps inside the reserve, as well as insects and things. And last year we did have not a mouse plague in the reserve, but get a boom of native mice after rains, and there was definitely plenty of mice around. So I suspect last year they were just chowing down on mice as well. But again, I'm hoping by going through that, I hopefully see where all those other the threatened species in the reserve fit in and how much they've been eaten. Hopefully we can show a bit of a story of what's been going on in terms of their numbers as well from the quoll diet. And the last kind of thing I've been doing is catching a bunch of quolls and putting GPS collars on them to try and work out where, how they're hunting and where they're hunting and what kind of animals they might be hunting for. And so it's kind of easier said than done. So I've set the collars to get like really high frequency fixes. So every 5 minutes at night, I get a GPS location, and then I have to go track them down in the field, and I download their data with a remote, and I look at, are there any points where there's lots of little sites close together, which means they've spent more than 5 to 10 minutes there. So you're assuming they're hunting or something, or looking for something. And then I go to all their sites and look for signs of other animals or dead things or animal burrows and things. And I've done a couple of quolls, and I haven't found any. Either they don't leave any traces of the dead animals, or they eat small things and they gobble them all up. But I'll show you some of the data. So these are 2 females I collared last year, and the girls generally have a pretty small home range, maybe like 1 to 2 square kilometres. So Tickle and Blue, she actually had a bunch of 3 youngsters in the den when I collared her last year. And when the quoll joeys get too big for mum's pouch, she will put them in a den, and then she'll come back and when she's weaning them, she'll bring food back to them. But I didn't record her, I guess, bringing any large prey items back to the den. So I think she'll still kind of fattening herself up to feed them. But what's cool is this is some of her data. Most nights she would leave the den, go south to this carcass dump, and we recorded her. It's just the black thing under her chin is the collar. Recorded her using the carcass stump, getting a free feed, and then she kind of do a bit of a loop and head out here where there's lots of mice burrows and things, and then head back at the night. So that was pretty cool to see and document. So the girls have been pretty easy to do that with. The boys, on the other hand, they like to go walk about and hunt for girlfriends. So this is a boy who pretty much most nights he would just do laps to the reserve, and it meant it was tricky to track down, and then I couldn't really get to where he'd been hunting until a few days after he'd been there, because by the time I found him, I'd only find him every couple of days. And then Carlton, this naughty boy, he actually jumped outside the reserve and went up here where there's this is the cat paddocks where they expose them to levels of cats, and then that's the outside the reserve there. So he, Quoll, the Arid Recovery has other issues with quolls, particularly the boys jumping over the fence, which means the ones that stay inside, a couple of males that do stay inside, they disproportionately responsible for being dads. There's not a lot of genetic diversity for boys inside the reserve because lots of them jump out, and then the ones that stay inside get to all the pick of the ladies to themselves. So they're a bit harder to study, and the males are larger, so you might think that they hunt larger prey, because quolls can take down things as large as them, so they definitely can take down a bettong that's 1 to 1.5 kilos. But yeah, that's kind of what I've been looking at, but yeah, it's kind of still ongoing, and I'm out here this month to try and catch another bunch of them and see what they're hunting. So in terms of all what everything means, I'm hoping we've kind of found that, that by exposing some of the animals to quolls, particularly the bettongs, that they change their behavior in response to being exposed to a predator, which is pretty cool. The vegetation is still kind of ongoing, so we need a bit more time to see what happens after the drought. And then looking at that scat data, that's going to help, I guess, paint a picture of how, in that gap where me and Mel weren't here, how much they ate the bandicoots and stuff, and the other animals that they're a bit harder to monitor as well. And then globally, hopefully all this kind of research will add to what we know about predator reintroductions around the world, and will add to that literature to, I guess, provide more concrete evidence of what does or doesn't happen when you reintroduce a predator. And then here at Arid Recovery, I'm hoping to produce a little scorecard to say which of these threatened marsupials that we reintroduce are kind of safe from quolls. So I think already I worked out that bilbies are pretty good. They're pretty hard to catch. I wouldn't bother eating one if I was a quoll. The bettongs and bandicoots, we've kind of shown that they're not going to go extinct so far, but it depends how. So I still need a bit of time to see how those populations respond again after the drought. And the bandicoots, because they're a bit smaller, we think that they're still around, but they might be a bit picked on a bit more by quolls. And the cygnet rats, unfortunately, got bigger problems. Unfortunately, because they nest above ground in these kind of structures that they build themselves, a lot of them got wiped out during the 2019 and 2018 heat waves, and so they're kind of not doing that well, and they need the managers here need to address, kind of make cooler homes for them so they can keep surviving. But the quolls, are their biggest problem at the moment. And then there's some quolls coming to some fence reserves near you very soon, or a bit far west in New South Wales. But there's future reintroductions of quolls planned for wild deserts, and at least one of the new reserves that national parks are building out near Cobar. So I'm hoping to give those managers some of my research, and hopefully they'll listen, but give them a quality tick of approval and say, yep, you can reintroduce quolls, and the species that you have there will be safe and they can live happily ever after together. So just like to again thank the Off Research Grant for helping to fund some of my work, and definitely makes a big difference having the funds to support all of my travel here and fuel in the field, and as well the Australian Research Council funding a lot of stuff with that, looking at the behaviour and granularity of those animals. There's lots of other things going on here in that space, and my work's just a small part of that. So that's very exciting as well. Are there any questions?
Anybody out there like to ask Ben a question?
It's Karen here. You were saying that the quolls are able to get out. These are cat-proof fences, I assume. How are they managing to do that? And are they actually managing to get back in again? Or is it a one-way street for them?
Very good question. So I'm just going back to the start. I've got a photo of a fence. So the cat-proof fences, again, to stop things getting in. If you're a quoll on this side, you can hop out. So right now it's kind of, there's too much fence for them to kind of double proof on both sides to stop the quolls getting out of the reserve. And so we have traps outside the reserve that occasionally when quolls get trapped in on the outside, we can put them back in. Hopefully they stay in, but there's some repeat offenders who know if they get trapped, they get kind of free food and bed for a night, and they just keep jumping in and out. But by themselves, last year, Macchiato, which is one of the boys I collared, another student was tracking him earlier in the year, and he had jumped out of the reserve, and we thought we lost him because he was a couple of kilometres away from the reserve, and then somehow he got back in. So when they're small, some of them they've seen can kind of squeeze through some of the netting if they really want to. So maybe he got back in before he was too big. But yeah, as a general rule, once they're out, unless we kind of catch them and put them back in.
Okay, thanks.
Okay, anybody else? Other questions?
Yes, Kim,
Ben?
I'd love to ask questions. I do.
Yeah, go ahead, Deb.
Deb, Andrew, thanks, Ben. Terrific. So the numbers of quoll from what you put in to what are there now? So has there been a significant increase in their numbers?
Yeah, it changes year to year, and depends who's around to look at them. So there are some other students kind of worrying about doing similar things to what I guess Dympna's doing, looking at their biology inside the reserve and their dynamics and things. So they probably in 2019 was their best year, and they probably got up to like 30 to 40 quolls, but then there was a bit of a lag where people weren't monitoring them and catching them and stuff to put them back in. And so there's probably, we don't really know. We're doing a lot of like camera trapping and stuff at the moment to try and work out who's who, and because you can look at their spots and put it through a computer, and everybody has a different spot pattern, just like our fingerprints. So that will kind of tell us what's going on now. But we reckon maybe between 10 to 20 adults consistently living in the reserve. So we find babies every year, but they're just kind of gone a bit. It's kind of because there hasn't been people properly tracking them down to monitor them, like in that first year when there was someone here full time doing that. And then we think maybe to 10 to 20. So they do need a bit of management to keep them in here and keep the population going. Whereas the Flinders, they've done great in the Flinders because it's a bit bigger area for them to get out of. There's about 70 quolls down there now in the Flinders, I think.
And how big an area is this reserve? Like, so how many quolls per hectare, or how big is, if you divided it up, say?
Where the quolls have access to is about 45 square kilometres. And so my math's not that good in terms of hectares, but you're probably looking at about one quoll every 2 square kilometres. Okay.
And is that comparable to this home range sizes they get in southwestern WA, or is it bigger?
It's kind of comparable. That I know, like the girls, they show a similar trend. Like the girls have a small home range, the boys go roaming. So there's definitely, I think it's pretty similar.
Yeah, terrific. And have you got predators of the quolls themselves as well?
Not really. Similar to what Dympna mentioned, if some of them are silly and are out too late in the morning, like sometimes maybe they think they've lost a couple to wedge-tailed eagles and things, but they're too big for goannas to eat. They're too big. We don't really have any big pythons here. And so, it's only if they jump the fence and run into a cat or a fox that's a problem for them. But inside the reserve, they're pretty safe.
There's a question on the chat line, Ben. Do you need to do genetic testing for the feces to find out what the quolls are eating of prey items that have already been digested?
Yeah, it's a good question. Technology, it's not quite there yet to do DNA testing of specific creatures. But with the scats, what we look for is for hard-bodied things, so scales and bones and stuff, but particularly the hair. So insects and stuff come up in feathers, and so it's kind of tricky to work out what feathers birds are. But with the hair, you can actually cut them under a cross section and look at them under a microscope, and every animal has a different kind of pattern of hair cells. So that's the way that we look at, which is the standard way it's been done for a while. But there's new technology coming, but in order to kind of look at like the DNA and stuff, you have to collect like everything that it could possibly eat and then send that to the genetic people, and they have to work out what's what and then match it up. And it's getting there one day. I think wildlife research will have access to that technology, but it's not kind of there yet right now.
Okay. Any last question for Ben?
How do I do it? Just talk. Would dingoes be their only natural predator? And do you have any dingoes, in your enclosed areas?
Yeah, I think dingoes would have been the next largest predator. Maybe larger pythons, like again across their range, they probably do come across bigger carp than the historic range. They would have come across like maybe bigger wimas and Murray-Darling carp pythons and things. But here at the reserve, we're kind of right on the dingo fence. So the dingo fence runs through the middle of the reserve. So south of there aren't really any dingoes outside the fence. North a little bit. About 15 years ago, they first put dingoes into the dingo pen, but put a pair in, and they kind of learned that they didn't do too well living in there. Like they kind of, as a big predator, they kind of hunted too much stuff, so they only kept them there for a little while and then let them go. But most of the work in there is with foxes and cats instead. It's just too small an area to kind of keep a captive population of dingoes because they have such big home ranges.
Right. Thank you.
Okay. All right. Well, I think we're just about at that time. That's fantastic, both of you. Thanks, Ben, and thanks, Dympna. But I'd like to call on Deb Andrew now to give you a formal vote of thanks. Thanks, Deb.
Hi there, Ben and Dympna. Thank you for fascinating and really exciting talks on your work. It's so exciting to see young people looking into these questions and doing work that's looking to rejuvenate our fantastic native fauna, and having a bit of a soft spot for Dasyurids and planigales and tiger quolls. I'm really jealous of the fact you're out there doing all this great field work. So we really look forward to seeing what your findings will be, and it's a great privilege for us to support you in doing your postgraduate work. So well done, and we're going to be looking forward to seeing western quolls and mulgaras and bilbies and bettongs out in the arid zone free ranging. Well done. Thank you.
Yep.
Yeah.
Thanks for having us.
Thank you.
I'm unmuted. Yeah. Yes, thanks again, and thanks for your time. It's not easy presenting when the internet's dropping in and out, and preparing it out there in the sticks. So yeah, really appreciate it. It's fascinating work. So we'll move on to the general business section of our meeting. To both of you, you're free to stay on if you want to hear all about the fascinating things that we do here in Sydney, or you may want to go off and finish dinner or have a drink or something. Leave it up to you. Happy for you to stay on and participate if you want.
Thanks, Kim. I might head off. I'm going to get out and radio track some of these bats, but thanks so much for having me. And yeah,
Great.
Thanks again for always.
Thanks, Dympna. We'll see you next time.
Thanks. Bye.
Okay. Oh, and just a reminder to everybody that most of our talks this year and last year are on our YouTube channel. So just scroll down and click on the button on the website homepage and you should get to it. And if you subscribe, you won't miss out on future videos because you'll get an alert. Okay.